Hello. Okay, so this is going to be the first part of your lab. So in place of taking a midterm, you're going to do a formal lab write-up. So the lab that we're going to do for your midterm is known as the analysis of food dye in beverages. Okay, so just a little bit of background because there's some information in here that we have not talked about yet, and that's fine because it's pretty straightforward. So let's go ahead and see what we're actually going to be doing. The objective is to use spectroscopy and graphical analysis to determine the concentration of food dye in a sports drink. So the concepts we're going to be working with is spectroscopy, which I'm going to talk about, absorbance versus transmittance, wavelength, Beer's law, and solution concentrations. Okay. Color tells us a lot of information about the composition of a solution. Okay, it can tell us quantitative information and it can tell us qualitative information. Qualitative analysis will tell us information about the nature or type of the compound. Okay, so these are things like physical appearance, things like that. Where quantitative analysis actually gives us numerical data for the amount of something within that compound. So what is spectroscopy? Spectroscopy is the study of the interaction of light and matter, okay? And we use a spectrophotometer to analyze the absorbance or transmission of light, okay? So what spectroscopy ultimately does is if we have something and we shine a light through it, okay, we ultimately can get a readout about what is actually inside of that substance, stuff that we necessarily could not see with our own eyes. Okay, so here's what we're looking at when I talk about a spectrophotometer, okay? So this is the system under the study, okay? And we'll look a little bit more what this actually looks like. This is the stimulus. So in our instance, it's going to be light. And what's going to happen is the solution is either going to allow the light to come through or not allow the light to come through, or let some of the light go through, some of the light not go through, based off of what is inside of that solution, okay? And however much light can go through is going to ultimately give us the response that we want. Now, when we're talking about this, we need to realize that light is on our visible light spectrum, on our electromagnet spectrum, okay? And we'll talk a little bit more about that when we get into the idea of color. So we're going to use glass cuvettes, okay? A cuvette is this guy right here, okay? This is the system under the control. And this is what we call a sample cell, okay? Because what we're dealing with is a solution. A solution is a liquid. And so I can't just inject liquid into my machine. I have to have something that holds the liquid in order for the light to pass through. And that's what this is. This is a cuvette. We also have, or it also uses a diffraction grating called a monochromator, which spreads the beam of light into the light's component wavelengths. So ultimately what this means is the desired wavelength is then focused onto the sample cell, so this guy, to detect any absorption or emission by light of a substance in the sample. So we're going to look at something specifically to that wavelength of that sample so we can actually get a really nice transmittance or absorbance. Okay, so let's talk about that. And this is kind of what you've been reading about in chapter 6. The absorption of visible light by a substance is a result of an electron transfer. Okay, the reason why we can see things is because electrons are moving up and down within their atoms. This ultimately means is that there's a promotion of a ground state electron to a higher energy level. So let's kind of look at what's happening here. So if this is the nucleus and we move out, right? If something moves up an energy level, it absorbs energy. Okay, so if I'm moving from one energy to another, I'm getting more energy, okay? And then as I fall down or I fall back down, so like right here, that's when I get the emission of the light, okay? So energy increases as you move out past the nucleus, okay? So if you move up, you get more energy. If you move down, you get less energy. All right, so let's kind of talk about our food dyes, what we're going to be looking at. The Food and Drug Administration, also known as the FDA, only has approved 7 unique dyes for use in food, drugs, and cosmetics. So the one that you're looking at right here is FDC number 1 blue, okay? You'll notice that it has a lot of double bonds within the structure, okay? That means that's why we get that very deep, vibrant blue color, okay? A solution that contains this dye under normal white light, it appears blue, okay? This is the color of light transmitted. So this is the color coming off of the sample into our eyes, and that's why we see blue. However, the color of the wavelength of light that goes into the sample and doesn't come back to our eyes, so the stuff that's being absorbed, is going to be complementary to the transmitted color. So whatever I see blue, the opposite is going into the sample, so I don't see it. And in this case, the opposite of blue is orange. So it's like orange, yellow, and red, okay? So the reason why I see blue is because orange, yellow, and red is absorbed into the substance, okay? So light goes into the substance, blue comes out, it's transmitted to my eyes, red, orange, yellow is absorbed into the sample. So like I'm saying, a color wheel provides a useful tool for identifying the colors or wavelengths of light absorbed by a substance, okay? So the blue solution absorbs yellow, orange, and red light. Therefore, we would expect to see the visible spectrum of FD and C blue between the 580, which is over here, to the 750, okay? So basically what this is telling us is the reason why we see blue is because these are absorbed. And in order to get the perfect peak or visible peak for blue, we have to look at the wavelength that is opposite it. Okay, so let's talk about Beer's law a little bit. So the amount of light absorbed by a solution depends on its concentration, as well as the path length of the sample of the cell. So for instance, we're using a cuvette, and most cuvettes are 1 centimeter long, so that's the path length. And then we deal with this constant known as epsilon. And this equation is just the molar absorptivity coefficient. It is consistent for each substance you're talking about, okay? It is just a constant, and if we remember, constants are given to us in order to cancel out units. Okay, that's it. Thank you.